everyone, and welcome to another interesting episode of The Doctors. My name is Dr. Zuni. I'm an oncologist from California and basically treat cancer patients along with doing research on how cancer could possibly be cured.
Greetings all, boy am I excited to be on this segment today. My name is Dr. Munn and I am a neurologist from Chicago. I generally deal with patients that have spinal cord injuries or deep brain injuries. Today's segment is on the negative impacts of cloning and how in some countries it is allowed to clone humans.
Recently, Dr. Munn and I have gotten together to perform research on DNA cloning.
More specifically, we have been looking into the negative factors of human cloning.
Well, let's start off with the introduction of cloning so our audience is fully aware of what it is and how it works. So, cloning is the production of an organism that is genetically identical to another organism. In other words, every single bit of DNA is the same between the two organisms. However, it is an unnatural process that takes place in a lab. When identical twins are born, it is a form of cloning naturally. There are several types of cloning that are used by scientists today. They include molecular, organism, and therapeutic cloning. Molecular cloning is the replication of DNA fragments by the use of self-replicating genetic material, such as a bacterial plasmid. The DNA of interest can then be propagated in a foreign host cell. Duplicates only individual genes of an organism, mainly used in medical research laboratories all across the world. This technology has been around since the 1970s and has slowly become very common. So basically, plasmids are self-replicating extra-chromosomal circular DNA molecules distinct from the normal bacterial genome. They were used by the Human Genome Project researchers to copy genes and other pieces of chromosomes to generate enough identical material for further study. Then comes organism or reproductive cloning. So this is the process of making an identical copy of an entire organism. It's accomplished by the process called somatic cell nuclear transfer. Last but not the least is therapeutic cloning. So it involves the cloning of human embryos for the production of stem cells. The embryos are eventually destroyed in this process. There are many ethical and unethical reasons for cloning. In our point of view, cloning is unmoral and unnatural, and therefore should be prohibited for any use. Recent statistics about human cloning. So according to Health Canada, the assisted human reproductive legislations prohibits human cloning using any technique. As stated in paragraph 5, these are direct quotations, so no person shall knowingly create a human clone by using any technique or transplant a human clone into a human being or into any non-human life form or artificial device.
I couldn't agree more, Dr. Zuni. Cloning should stay prohibited and all over the world. I can't believe some countries actually allow human cloning. Over time, cloning has highly developed though. In the 1880s, Professor Weizmann at the University of Freiburg proposed that with each cell division, the genetic information of the two cells depreciate. And Wilhelm Roux proved Weizmann's theory when he observed that only half embryo developed when he destroyed one cell of a two-cell frog embryo with a hot needle. Hans Spemann disproved Roux and Weizmann's findings in 1901 when he found that embryo cells in their early stages contain all genetic information. Spemann came to this conclusion when he split a two-cell salamander embryo into two parts, and he successfully grew them into two complete individual organisms. In 1902, Walter Sutton hypothesized that all genetic information is contained in chromosomes within the nucleus, which we all know of today. In 1938, Spemann publishes results from his nuclear transfer experiments and explains in his book Embryonic Development and Induction, transferring one cell's nucleus to an egg without a nucleus provides the basis for cloning experiments. In 1962, John Gurdon of Oxford University clones a South African frog from the nucleus of a differentiated adult intestinal cell. Differentiated cells are cells that are part of a specific tissue or organ which produces certain proteins because of the specific genes that are activated in that cell. This is what distinguishes a liver cell from a skin cell, for example. John Gurdon used differentiated intestinal cells, nucleuses, to clone the South African frog, which was very successful. In 1964, Stewart proved it was possible to clone from differentiated cells, also supporting his theory. In 1969, James Shapiro and Jonathan Beckwith of Harvard University isolated the first gene. In 1983, Carey Mullis invented PCR, polymerase chain reaction, allows for the rapid replication of fragments of DNA. In 1990, as we already mentioned before, the Human Genome Project was started. In 1996, Ian Wilmot and Keith Campbell cloned Dolly the sheep using differentiated cells as well, which was also thought impossible because the cells have already been specialized to a specific body function. This is also known as reproductive cloning using somatic cell nuclear transfer. They cloned Dolly by extracting a mammary gland cell from a Finn Dorset sheep and extracting an egg from a Scottish blackface ewe. They removed the nucleus from in the egg cell, but leaving all other crucial components of the egg cell in the egg to help develop an embryo. The two cells from the Scottish blackface ewe and the Finn Dorset sheep were both placed beside each other and had an electric shock applied to them to help them fuse together. The mammary gland cell is now inside the egg. A second electrical current was also applied to help the egg to start cell division. They kept the cells dividing in a laboratory dish, letting it develop into an embryo, which was then placed inside a surrogate mother sheep's uterus, also a blackface ewe. 6 days later, after about 147 days, Dolly was born, identical to the Finn Dorset sheep. In 1997, President Clinton signs for a 5-year moratorium for the use of federal funds for human cloning. Clonaid, first human cloning company in the world, was created in 1997. The first human baby clone in the world was born on December 26, 2002, named Eve, by Clonaid. Eve was born to a 31-year-old American woman whose DNA was identical to the mother's DNA. 2001, scientists at Advanced Cell Technology Inc. announced birth of a clone baby ox named Noah, which then died shortly after the procedure because of an infection they claimed to be unrelated from the cloning process. Dolly the sheep develops lung cancer and arthritis and is put down in 2003. Clonaid has claimed to successfully clone more than 4 human babies in 2003 and provides human cloning services to this very day.
Wow. So Eve was the first baby that was cloned, and we have a tweet from the audience that we would like to answer right now. Dr. Munn, would you like to read it?
The question was from Lucy. How is the first cloned baby Eve doing now, and where is she? Well, the president of Clonaid, Bridget Bouslier, says Eve lives in Israel, and she had seen the baby Eve over videotape in 2003. But the parents had cut out all contact with Clonaid. Very suspicious. She also says Eve is healthy and has no differences from natural-born children. But no one knows if this is entirely reliable information since there hasn't been any evidence of her existence provided by Clonaid to the public. Many scientists also believe Eve is a hoax because they are unable to do any testing on Eve. We are not able to see her, nobody has seen any pictures, and now we don't even have contact with the parents anymore. Yep. Very suspicious.
Yep. So they're assuming she's fine, but we don't know if she was successful even though she was cloned. Very interesting. Seeing all these failed and unreliable attempts of human cloning, we can already predict that human cloning will not have a bright future. Here are several more issues associated with human cloning. So, human cloning can lead to several diseases. Cloned humans tend to have more compromised immune functions and higher rates of infection, tumor growth, and other disorders. Defect in genetic imprint of DNA from a single donor cell may lead to some developmental abnormalities of cloned embryos. Implementing chemically marks the DNA from the mother and the father so that only one copy is turned on. So another problem is that a number of cloned humans are born with birth defects. A possible cause for the defect in clones are the epigenetic changes. So apart from the DNA, the genome carries a large layer of information in the genome called the epigenome. Clones derived from adult cells do not have the epigenetic patterns found in a newly born, so they could carry defects.
Another negative impact on human cloning is on embryos. The process causes harm to the embryos and to the cloned person throughout their life. Since many embryos are created and tested for viability during the process, most end up being discarded or frozen for further use. The discarded ones are killed, which may be considered murdering.
Another downfall to human cloning is that it's considered to be a very unnatural way of producing humans. Many religious views state that cloning is an unnatural form of producing humans and is morally wrong when they can sexually be produced by two parents. It is also against the Canadian human rights to clone humans. So cloned humans may not have full human rights since they are just copies of others' DNA. It also creates a conflict with the societal values about human dignity, principles of human freedom, identity, and autonomy.
Those are all very valid points, and I couldn't agree with them more. Another con to human cloning is the high failure rate. It's inefficient, and more than 90% of cloning attempts fail, and there is no offspring produced at all, or it dies shortly after it is born. Reasons to why they fail is because the enucleated egg and the transferred nucleus may not be compatible. Another reason is an egg with a newly transferred nucleus may not begin to divide or develop properly. Implantation of the embryo into the surrogate mother might also fail.
Wow, so another downfall to cloning is that the technology may be harmful to the ecosystem. The more cloning that takes place, the less genetic variation there will be. So it negatively affects a diverse society because everyone will want to produce humans with perfect skin tones, eye and hair color, and so on. People will constantly try to make the best genetic versions of humans. It will interfere with the natural evolution of humans and impact the future generations. If humans are cloned to live longer, the population will not die off and more humans will be produced, resulting in an overpopulated society. There may not be enough animals to feed the humans, since most humans are carnivores. Well, the chain reaction will continue to impact our...
I'm a vegetarian. Anyways, that's another debate we'll have on our next segment.
Well, unlike people like her, there's obviously enough plants to eat. So the chain reaction will continue to impact the Earth as a whole, and it will eventually be ruined because of human cloning, over time obviously.
Another huge con to human cloning is obviously the misuse and taking advantage of cloning. People in power may clone humans with physical strength to form an army for the country. Such human clones are not able to make their own decisions about their lives and are forced to perform such tasks. The rich may start producing babies with ideal genes and use them as models, etc. Also, the rich people will have the power to create an unnatural ideal baby for themselves, whether it is modified to have a specific eye color like we mentioned before, or lacks a certain disease gene.
Wow, that's a very valid point. Lastly, we think that a problem with human cloning is that it's very expensive. Rather than spending money on such a technique, babies should be naturally produced by two parents. Bias to those who are not able to afford the treatment. So a lot of people who are able to afford it can have certain benefits, even though I'm totally against cloning, whereas poor people don't have the same advantages. Also, since the failure rate is high, the majority of the money spent on cloning is gone to waste. The same money can be used for a better cause, such as cancer treatment or cure, as long as it's research. Also, it's harmful to the economy because cloning is not necessary to survive, whereas the same money can be used to save an individual's life.
As you can see, there are many negative aspects to human cloning because of the many unethical and unnatural factors associated with it. Many people believe that humans should be produced through a natural process, which I couldn't agree with more. Rather than copying the exact genes of an existing human being, why not be original? Human cloning will not only affect the parent and offspring, but it will also affect the society as a whole in the long run. In the rare cases that human cloning proceeds successfully, the lifespan of a human can increase by resourcing to organ transplants from their clone. This will cause humans to become overpopulated and impact the food chain. The earth will become an uncivilized place.
Since only the rich and powerful people of the society would be able to afford this procedure of human cloning, they would be able to live longer in the sense that they would have an organ transplant ready whenever complications arise. Average to poor income families wouldn't be able to afford this process, leaving them with a shorter lifespan to those who have cloning advantages. Also, the topic of human cloning can be very controversial as different groups have certain beliefs towards it. Overall, this technology has the possibility to majorly ruin the nature of life. After numerous amounts of experiments and research, it is best to prohibit the process of human cloning.
Is it really fair to let the rich and the powerful maintain their statuses with the help of their clones while the average to poor income families become ruled out because they are weak?
Wow, now that's a brain teaser. What do you guys think? Well, that was the end of our show.